Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers, welcome to the TV learning series. Here we are again with Senior 5. We are moving molecules, we are moving ions, we are moving water. So the topic there is still under transport in plants, as you can check on the slide. And the lesson today, we are looking at 1, where do we get water from? And 2, how do we move it up through the plant? So our focus is on one section of the transport tissues, where we were looking at the xylem previously and the phloem as transport tissues, but today's focus is centered only on the xylem. So what are we supposed to achieve at the end of the day? 1, we need to have some bit of remembering, able to recall how the xylem is shaped. That's objective number 1. And where it is found in the tissues, where it is found within the organs. So if we are to look at the stem or root, where can we trace, where can we identify the position of the xylem? Second objective, that we need to check the features of the xylem. What characteristics does it have? If it is to allow water to move through it, is it fully equipped? Water and mineral ions as 2 substances being taken by this xylem. How equipped is it? What characteristics does it have? What special adaptations? And then we describe a very important command word, step-by-step process of an event. So if we are looking at mechanisms, then we need to describe them. We need to look at what starts first, followed by what. So an event is taking place. Question is where, and 3, how. Not why. The whys are not involved here. We don't want reasons as to why water is moving or as to how water is moving. We are only talking about how it moves, step-by-step process. So it's like we are writing an observation of what happens when water is going through the roots and then up to the xylem, just there. But a few things we need to look at. So now let's focus on the slides. What is the most important capture of the whole lesson? 1, we need to find the relationship, a positive one, with the structure as the xylem and what it does. So when we talk of adaptations, we are now looking at what features does the xylem have to be able to have full equipment, full design to allow minerals and water to go through. So the question now is, we have already seen these things. What does the image in the background remind you? That's our take today. Hollow. Do we have any gates, any barriers? Secondly, what about the linings? Oh, this looks like a beehive. Every ring is composed of lignin. Remember, this is an electron micrograph. Now, the walls that have got lignin deposition, the walls that have that chemical, that organic substance, which has to ensure that 1, hardening. So it's like compressed concrete, the enhanced concrete, where you have gravel as well as metal all put together to support structure. Now we need to start thinking, if every ring is made of lignin, then it means that the walls of the xylem are going to be 1, tough, 2, strong, and 3, no life in these holes. What does that mean? When water is moving through these tubes, it flows with pressure. The question is this pressure able to break down these tissues? And if it does, should there be a way in which the tissues are equipped to withstand these forces with which water moves through them? It's like a comparison of a water pipe, plastic, and a metallic pipe, and they are all allowing water to flow. There are situations where water pressure has always burst the pipes, and then water starts flowing all over because a water pipe has burst open. Or at home, and then you have to look for these plumbers to come and then again fix these things. So the same applies to plants. Water passing through these vessels flows with pressure. So there has to be a force to counter. And this will be brought about by the nature of the vessels, lignified, date, enable one to allow bursting, but also this lignin cannot react with minerals. So we talk of a pool of transpiration, then this is exactly what we are looking at. That's what we are capturing today. Going to the diagrams, again, we need to recall where are they positioned, what do they look like. If we were to draw them, can we draw it exactly the way the microscope displays it? Definitely no. So if we were to draw, what do we exactly have as our representation when it comes to these tissues? That's one thing you have to ask yourself, which takes us to this term, a plan diagram. So now when we look at house designs, do they show the bricks? Or they only show the position of the window? They show the size of the wall, but they are not telling us the bricks and the way they are arranged. So that's a house plan. The same applies to this, is our plan diagram. So a student is drawing this, but observing what we see here in these images. The cells are here shown by the microscope, but in the student's diagram, the cells are not indicated. But pay attention to this. The region where these cells are, which is here, is represented this way. So it's just a plan. A plan diagram shows the region where the cells are located, but does not show individual cells. Because you don't need to draw each cell. So the image here is trying to remind us two things. One, where do we find the xylem? Where do we find the phloem? Where is the cambium? We don't show cambium cells. But in our micrographs, these cells are present. Check the xylem and check the position. Check the cambium, check the position. Look at the phloem and check the position. The purpose for bringing this back here is to remind us, one, the location of these tissues. Our focus, remember, is only on the xylem. Correct? Yeah. So now, if we are to look at the xylem drawn as a simple diagram of a student showing the plan, and here in the micrographs, we are able to see them as hollow. Remember, the characteristic is that they are hollow. There is space in between to allow a continuous flow of water. That's the capture from this slide. We are showing the xylem vessels and the phloem. Location, xylem towards the inside, phloem towards the outside. Xylem vessels can be also identified by their large empty lumen and the thickened walls. So we need to check in the simple drawings, in the simple images. Do we see something like a hole? And around it, how thick is the lining? So that we're able to say, okay, yeah, that is the xylem because I can see there is space inside, and the region as a circle tries to show the thickness. So in this section, as a paragraph, two things we're picking from there. Can we identify the structure? Can we differentiate it from the phloem? Now, coming back to our water pipes, whether metallic or plastic, whether straws we use to drink juices and sodas, how is the scientist thinking to be able to design a water pipe that is not able to burst, or a straw that is able to withstand the suction pressure as you drink and then it doesn't collapse? That's our thinking, which takes us to the structure of the xylem. When we look at this one hollow, remember, when we say structures, a hollow lumen is not a structure, it's not a part, it's space left behind by the structures. So what structures are we looking at? The cell wall. What else do we have to look at? We have a cell wall. Sorry, we have this as the cell wall. Another structure would be the spiral lignin. But the label here we have is only space. Something else we need to remember, that these are initially cells during development. That's why we have walls. That's why we have the word cell wall. During development and their formation, initially we have cells that actually have even ends. But where have these ends disappeared? Breakdown, making one cell connect to the other, but this time as hollow. So it means, therefore, from here to here, there is continuity, because the plates break away, creating a stream, continuous flow. Pits are also space, connections between cells. But when you look at them in their detail, as we shall see, or as you have seen in plant structures, plant cell structures, you have tubules and microtubules filling this region to help cells communicate with each other. The same applies to xylem. These small gate-like structures do allow one xylem to communicate with another. So water can flow, therefore, from here, move in, climb, reach here, connect to the other. Which makes us start thinking, the thickness of the tree, bottom line, and then the top where it's getting smaller. How is that possible? If we have 100 tubes down and it's this big, do we also have 100 tubes here? How can 100 be here and again 100 be here? Assume that this is my stem. 100 are here. Can I have 100 here? So how do they link that towards the end, in the branches and the extremes, we still have water continuing to flow? That again takes you back to the way these are arranged in your senior form. So we are not going back to that. I'm just bringing it up for you to be able to connect. What else do we have? the remains of end walls before they broke down. I love the language. So they were there before. Remains of end walls, meaning that from here to here was a cell connecting to another one down. So the walls broke down, creating space, creating a lumen, allowing a continuous flow. Like a water pipe that has a hole inside and there's nothing to stop this water from flowing. So they are arranged end to end to form continuous vessels. Reduction of the walls between the cells in the vessels would make it easier for water to move between cells without being stopped. Lignin, complex fibrous organic polymer. 1, strong. 2, cannot react with minerals. And so they will flow through without being consumed. I love that. Someone gives you a lollipop, take it home without you getting it out of the paper and consuming it. So lignin, 1, is deposited, 2, makes the wall tough, 3, makes the wall dead, and 4, does not react with minerals. All those features to allow the xylem to be able to transport water. 1, don't consume the water. 2, don't react with the minerals you're taking. Right? So, the uptake us. We are now going. In the usual way, we all know, okay, every time we water, when we were young, we used to think that the proper way to provide water to the plant is to put it on the leaves, so it gets in directly. Little did we know that actually, when you're watering the plant, it has to first go to the ground. So the question is, which is the best irrigation system? Sprinklers all over, or underground, buried small pipes to push water directly into the soil? So start thinking. So here we are, water and minerals outside. Foods, carbon dioxide, light, sugars are made down. Remember that our focus today is only on the blue arrows. So how does water get out of the soil into the plant? Do we have small pipes to collect water like no. So we have now the chemistry coming back. We now have the physics coming back. So osmosis, you've seen it before. Now here it comes. The definition helps us to understand how water goes in. What are we talking about here? A high concentration of solutes, maybe the minerals, the sap content in the cells. So that water outside has more potential, has more ability to move in. What are we talking about? We have the soil with minerals. We also have the root hair cell on the epidermis of the root, also with minerals. Interestingly, like you mentioned before in your senior 4, active transport will always keep the minerals high in concentration in this cell, causing this cell to be like thirsty in a way, thirsty. So, it will renew it. Meaning, amazi azava hano aho ari menshi across the cell membrane into this cell by osmosis. High water content outside, low water content inside. So by osmosis, water moves from high water potential to low water potential. When we talk of water potential, we are looking at the ability, the force with which water can easily move from where it is a lot to where it is less. Water potential. Ubasha bw'amazi kuva aho ari menshi aja aho ari make. In the simplest words, high water potential, high ability region with water to go to where there is less. We are using the word osmosis because we have to start thinking about one, physical, no energy consumption, cell membrane, partial permeability, and stuff like that. So, down the water potential gradient. High to low. Into the root hairs. Now, once here, the cell next to this is more thirsty, I'll use the quotes, than the previous one. So the cells in the cortex, the cells inside the root, have low water potential than the cells on the surface of the root. So in the same way and same explanation, water will move from the epidermal cells into the cortex cells, way through by osmosis, following water potential gradient until, boom, inside the xylem. Remember, we looked at this before. So I'm using the image to just give us an overview, an understanding. Once in the xylem, sasa, viewers bigatangira. If the pressure in the cells was pushing the water up here, what is going to happen once this water is in the xylem? Remember the rings, and now we have water ascending. So this is the uptake, this is the ascent. But we need to start thinking, while our focus was mainly on the xylem, how do we relate the xylem with water? Which takes us to a few things we need to check. Let's start with this image. This is a small section, the stick or a leaf, and the drops of water are clinging, they are hanging there, and they are not falling off. How does that happen? That very many water molecules keep together, and then other water molecules are lying flat and sticking to the surface of the stick, and they don't fall off. So, two terms. One, cohesion. Forces of attraction between molecules of the same kind. So water will keep water together as we see it. It flows together because the forces between the water molecules keep holding them together. What are we talking about here? The hydrogen bonds here keep this water molecule and this water molecule and this water molecule and this water molecule together. Even when they are weak, that when we boil the water, these will break. But at room temperature, when water is a liquid, this gives the strength within forces, and so water molecules will start pulling other water molecules via those forces. So that makes water kind of sticky within itself. It is the same force with which we are looking at the closeness of these molecules on the surface of water acting as a launch pad or a landing area for some of these animals, like you can check in the image. Now, when we go to capillaries, tubes, straws you use to drink sodas, have you ever made that observation? How you put a straw in water or a soda and you see the soda climbing up by itself and boom, it starts flowing. Is there any force that is pushing the water to climb up and move? So this now takes us to the second force, capillary actions, where we have cohesion, so the water molecules pull each other and we go, but also we have other forces, adhesion, attractional forces between the different. The stick has its own surfaces different from the water molecule, but now they are together and they are different, adhesion. Rushing to this now, where we now look at adhesion, like we said, our water pipe, water molecules are pulling each other, as you can see, if we can keep flowing as we climb. But also, the water sticks to the ends of the walls, like we just mentioned here before. So these two, adhesion and cohesion we've just mentioned, plus capillary, an action caused by combination of adhesion and cohesion, allowing water to move through the xylem vessels. Tiny, they are very small, like capillary tubes. Very helpful in movement of the plant, water in them, but the question is, how far? How tall would the plant be to only use these forces? To say, only for shorter plants. When plants get tall and taller, as you can check this image, compared to a bean plant, I mean, the difference is so big. These three forces, adhesion, cohesion, capillary, and root pressure, may be enough to cause water to move up in shorter plants. But as plants get taller, we might need something else, which we are yet to see. So, the flow, like we just talked about, the forces, in relation to the pictures we've seen, this is a wrap-up. The first image should be able to show you where we can tell, do we have a xylem, do we have phloem, what is the location? How do I compare the two? The image down here, which we are wrapping up with, pits to allow communication between cells, between xylem, as you can check, and the upward movement, one-way flow. Adhesion, water molecules stick to the surfaces. Cohesion, one water molecule will pull another as they go up. If they reach here, then they won't fall back. Upward movement through the xylem generated by transpiration pull. What are we talking about? Here, as water is lost, more will come up. Once in the stem, these forces will cause the water to keep moving. So to wrap it up, when transpiration is to take place, it means, one, first lose the water, allow more water to come in. For any uptake, there has to be a loss, first of all. So today, as we wrap this up, we started from here, moved into here, now here. The next session, we shall be in the leaves, and we get rid of the water. With that, we come to the end of this lesson. Thank you so much for following. See you another time when we go into how water is lost through the leaves. Thank you so much.